A very good morning to my dear students of class 11 science. Welcome to biology class. Today we'll begin with a new chapter, photosynthesis in higher plants. We have learned about photosynthesis in class 10 also. So today in detail we are going to learn about this topic. Okay, so what do you mean by photosynthesis? Generally we have learned that the green plants which contain the pigment chlorophyll, they used to trap the solar energy, they used to trap the carbon dioxide and water, and mainly they used to produce the food particles, that is the glucose, right? And we used to call it photosynthesis, that we have learned in class 10 definitions. And mainly the plants that contain the chlorophylls used to perform the process of photosynthesis, and those plants are called autotrophs, and they have got autotrophic mode of nutrition. So now in details we are going to learn about this chapter. So what do you mean by photosynthesis? So it may be defined as the synthesis of carbohydrates by the green plants or by the green organs of a plant in presence of sunlight from carbon dioxide and water taken up from the air and from the soil respectively. A little bit of different definitions, but the same. Okay, so we can say that during photosynthesis the green plants that contain the pigment chlorophyll, okay, in the presence of sunlight and carbon dioxide and water, they used to produce the food or the carbohydrates. Clear? And in this process the oxygen will be evolved as the byproduct, so oxygen will be released into the environment. Now where does this photosynthesis takes place? Photosynthesis takes place in the chloroplast. So chloroplast is the site of photosynthesis. Now photosynthesis may also be defined as the capture of photons, the unit of light, and the conversion of this light energy into chemical energy. Okay, so chemical energy. So light energy will be converted into chemical energy during photosynthesis, and energy will be stored. Clear? Now the overall reaction of photosynthesis is that 6 molecules of carbon dioxide plus 12 molecules of water in the presence of sunlight and the pigment chlorophyll, the plant will produce the food or the carbohydrate or the glucose C6H12O6. This is the food molecules or the carbohydrate that is produced during the process of photosynthesis, where oxygen will be released as the byproduct into the environment. Okay, so this is the overall reaction of photosynthesis. Clear? Now during photosynthesis the green plants, with the help of chlorophyll, the green pigment bearing cells, produces or synthesizes carbohydrate from carbon dioxide and water and in the presence of sunlight. Okay, now carbohydrate produced by the photosynthesis stores the chemical energy for the entire living world. That means those carbohydrate or those food molecules which are produced during photosynthesis, they contains the energy. Okay, it contains the energy for the entire living world on the earth, because those food particles which are being produced by the plants will be consumed directly or indirectly by the other plants or by the other animals. Okay, so therefore the green plants which perform the process of photosynthesis, they are called autotrophs, and their mode of nutrition is called autotrophic nutrition. Now what is the significance of photosynthesis, or what is the importance of photosynthesis? The most important thing is that during photosynthesis the sun's energy is trapped by the autotrophic organism, that is by the green plants, and it is being converted into food for all the living organism. Without the green plants we will be devoid of food materials. Next thing we can say that during photosynthesis oxygen is liberated or is being released into the atmosphere. It's a natural phenomenon or natural process where the oxygen is released into the environment. Next thing we can say that photosynthesis is only the method to manufacture food from the inorganic raw materials. Okay, next one we can say that it will decrease the concentration of carbon dioxide which is being added to the environment by the process of respiration and by the process of combustion. Okay, so these are the main significance of photosynthesis. In terms of the global warming criteria or the problems, we can say that photosynthesis plays an important role because it will maintain the balance of oxygen and carbon dioxide in the environment. It will release the oxygen in the environment and it will take the carbon dioxide, because carbon dioxide is one of the raw materials for photosynthesis. Okay, now next one we will discuss about the development of knowledge about photosynthesis, or we'll discuss about the historical background of photosynthesis. So we have got the scientist called Joseph Priestley. It's in the page number 13.3. okay, we'll see the contribution of few of the scientists for the process of photosynthesis. So Joseph Priestley, he put a burning candle in a closed glass container. Even you can see in the figure, okay, figure 14.1. so he put a burning candle in a, this is the burning candle, okay, in a glass container. Now he found that the air inside the jar had changed and would not allow the candle to burn anymore or a mouse to live in it. That means in a closed glass container, okay, Joseph Priestley put a burning candle and he placed a mouse. Okay, so when the candle was burning inside the glass container, that burning candle usually changed the quality of the air inside the glass container. Okay, and it did not allow the candle to burn continuously, and the mouse which was placed inside that glass container also was dead. Clear? But in the second experiment, what he did, that he put the burning candle, he put the mouse inside the glass container, but he placed a twig of mint in that glass jar. Okay, he put a twig of mint. Now you observe that the air would neither extinguish a burning candle, nor was it all convenient for the mouse which was put into it. That means in the second glass container, where he put the burning candle, where he placed the mouse and a twig of mint, the candle used to burn continuously and the mouse was alive. The mouse was not dead. So from this experiment he concluded that plant, that means the twig of mint which is placed in the second glass container, used to purify the air which had been imbued by the burning candle. Okay, so Joseph Priestley, what he had done, in the first glass container he simply put the burning candle and the mouse. The burning candle used to change the quality of the air inside the glass container, due to which the mouse was dead. Okay, because the air was not being purified in the first glass container. But in the second glass container he put the burning candle, he put the mouse and a twig of mint. Okay, so in that particular glass container the candle used to burn continuously and the mouse was kept alive. Why? Because that twig of mint, or the plant which was placed in the second glass jar, used to purify the air which was being made imbued by the burning candle. Okay, so this was the role of Joseph Priestley in the process of photosynthesis, that the green plant used to purify the air. Clear? Now the second one we go learn about starch. He discovered that the product of photosynthesis was starch. Engelmann, what was the role of Engelmann? He simply write this one. Okay, he discovered the effect of different wavelengths of light on photosynthesis. This we will discuss afterwards. Okay, we will go through the light harvesting complete of photosynthesis and we will discuss it in another class also. So Engelmann used to discover the effect of different wavelengths of light on photosynthesis. Blackman, he discovered he proposed the law of limiting factor. Warburg used unicellular green algae to study photosynthesis. Van Niel demonstrated his studies with purple and green cell bacteria that photosynthesis is a light dependent reaction. Okay, so Van Niel proposed that, or demonstrated that, photosynthesis is a light dependent reaction in which hydrogen from an oxidizable compound reduces carbon dioxide to form sugar. So these are the name of the few scientists and their contributions which you have to go through this chapter. Clear? Okay, now next one we'll learn about the chloroplast, the site of photosynthesis. In class 10 we have learned about the structure of chloroplast also. Clear? So chloroplast is the site for photosynthesis. Clear? And they are present in green stem, sepals and leaf. Now let's see the ultra structure of chloroplast. So chloroplast is mainly a double membrane structure. Okay, double membrane structure. You can see the outer membrane and you can see the inner membrane. So chloroplast is bounded by a double membrane envelope. Now in the interior part, or in the inner membrane of the chloroplast, we will see the proteinaceous colloidal matrix which is called the stroma. So we will find the stroma part, okay, inside the chloroplast, and we will see an elaborate system of membranous lamellae called the thylakoids. So we will see the stroma and we will see the thylakoids in the ultra structure of chloroplast. Thylakoids means simply the grana. Okay, so here we will see the stroma lamella and the grana lamella. Clear? Okay, now let's see the stroma part of the chloroplast. Okay, so the stroma contains DNA, RNA and 70S ribosomes. Okay, the chloroplast DNA is circular. Look over here. The DNA of chloroplast is circular and is naked and is also called the plastidome. Okay, so the stroma contains DNA, RNA, it contains 70S ribosomes. The DNA of chloroplast is circular and is naked and is called the plastidome. Clear? Now, what are the enzymes present in the stroma? Okay, and what are they associated with? So it contains the enzyme that will help in the Calvin cycle of photosynthesis. We will discuss it in the next class. Synthesis of photosynthetic pigments takes place in stroma. DNA replications, RNA transcription, protein synthesis will take place in stroma. Lipid metabolism and synthesis and degradation of starch. All these events will take place in the stroma. Clear? Now let's talk about thylakoids. Thylakoids lamellae, they run parallel in stroma and are divided into two types. Okay, grana lamellae or the grana thylakoids and the stroma lamellae or the stroma thylakoids. Listen very carefully. So thylakoids are mainly divided into two types. Number one, grana lamellae or grana thylakoids, and we have got the stroma lamellae or the stroma thylakoids or the Fred channels. So which one is grana lamellae? You can see over here. This one is the grana. Okay, so this is the grana lamellae. Even this one is the grana lamellae. Okay, these are the grana lamellae. And this one, which will contain the two grana, this is called the stroma lamellae. So this one is the stroma lamellae. Okay, which is connecting the two granum or the grana lamellae. Clear? Okay. Now let's see that the chloroplast has 40 to 100 grana. Okay, so there are about 40 to 100 grana, and each grana has about 10 to 100 grana lamellae. Okay, so thylakoid membrane possesses photosynthetic pigments. It has got pigment chlorophyll A, chlorophyll B, carotenoids, cytochrome B and F, ATP synthetase, and enzymes needed in photochemical reactions or light reaction of photosynthesis. So what the thylakoid contains? Thylakoid contains the photosynthetic pigment that will help in photosynthesis, that is chlorophyll A, chlorophyll B, carotenoids and cytochrome B and F. It also contains the enzyme, that is ATP synthetase and other enzymes that will help in the photochemical reaction or the light reaction of photosynthesis. And mainly the thylakoids are the sites for the light reaction of photosynthesis. So we have finished about the structure of chloroplast. So we have learned that it's mainly a double membrane body structures. We have got outer membrane and the inner membrane, and it's divided into the stroma and the thylakoids. Stroma contain the enzymes, either DNA, RNA, similar ribosomes. DNA is circular and naked, is also called plastidome. Okay, in the stroma part of the chloroplast, the Calvin cycle of photosynthesis, there will be synthesis of photosynthetic pigment, there will be DNA replications, protein synthesis. Okay, there will be RNA transcription, lipid metabolism. So all these events will take place in the stroma part of the chloroplast. Then we have got the thylakoids, which run parallel in the stroma and divided into the stroma lamellae and the Fred channels, or the, we can say, grana thylakoids. Okay, so we have got grana lamellae and the stroma lamellae, which are the part of the thylakoids. Clear? Now these Fred channels or the stroma thylakoids will connect the two granum or the grana thylakoids that we have learned. Then we have learned that the thylakoids contains the pigment like chlorophyll A, chlorophyll B, carotenoids, cytochrome B and F. It contains the enzymes like ATP synthetase, which will help in the light reaction of photosynthesis or the photochemical reaction of photosynthesis. And that thylakoids are the sites for the light reaction of photosynthesis. Now, since we have discussed about the pigments of photosynthesis, what are the different pigments which are involved in photosynthesis? We generally know about chlorophyll, right? Chlorophyll are the pigment that help in photosynthesis. But what are the different types of chlorophyll which will help in the process of photosynthesis, and what are the other pigments also which are there? Okay, so mainly we have got three groups of pigment which will help in the process of photosynthesis. We have got chlorophyll, carotenoids and phycobilins. Okay, one more time saying that there are three groups of pigment which will help in the process of photosynthesis: chlorophyll, carotenoids, and phycobilins. There are different types of chlorophyll. Okay, we have got chlorophyll A, B, C, D, and E. Clear? We have got chlorophyll A, B, C, D, and E. But out of them, chlorophyll A and chlorophyll B are the two main photosynthetic pigment. Okay, so out of A, B, C, D, and E, A and B are the most important photosynthetic pigment of plants that will help in photosynthesis, and they will absorb the wavelength of light. Okay, different wavelengths of light that we'll discuss afterwards. Clear? So remember that we have got chlorophyll A, B, C, D, and E, but A and B are the most important one that will help in the process of photosynthesis. Then even we have got one particular pigment called bacteria chlorophyll. Bacteria, they used to possess this bacteria chlorophylls. Okay, and even they have got this one called chlorobium chlorophyll. So bacteria, those that perform the process of photosynthesis, they have got bacteria chlorophyll and they have got chlorobium chlorophyll. Kindly remember this thing. It's in the page number 13.6. So bacteria that perform the process of photosynthesis, they contain bacteria chlorophyll and chlorobium chlorophylls, also called bacterioviridin. So kindly remember these terms. Clear? This is very, very important. Okay, now let's talk about the next topic, absorption spectrum and the action spectrum. What do you mean by absorption spectrum and the action spectrum? It's in the page number 13.9. Page number 13.9. Now, since we have just learned about the different pigment of photosynthesis, we have got chlorophyll, phycobilins, right? We have learned this one, carotenoids, and in chlorophyll we have learned that we have got A, B, C, D, and E. Out of this, A and B are the most important one. Okay, so different photosynthetic pigment, that will help in photosynthesis, they absorb light of different wavelengths. Clear? So different pigment of photosynthesis, they used to absorb light of different wavelength, and this can be shown in the graph. We can see in the page number, figure number 13.7 also, the graph that mainly shows the light which is being absorbed at different wavelength. This is the one. Clear? So here we are going to discuss about this term, absorption spectrum and the action spectrum. So what do you mean by absorption spectrum? So curve obtained by plotting the absorbed amount of light of different wavelength by a particular pigment is called absorption spectrum. So here you can see the absorption spectrum, or the amount of light or the wavelength of light that's absorbed by chlorophyll A. So this is the curve which shows the light wavelength of light absorbed by chlorophyll A. This is the wavelength of light absorbed by chlorophyll B, and this is the wavelength of light absorbed by the pigment beta carotene. So when we used to plot the wavelength of light absorbed by the different pigments, then when we plot it, we'll get a curve or we'll get a graph which is called the absorption spectrum. Clear? And what does action spectrum means? Go to the figure number 13.8. The curve showing the rate of photosynthesis at the different wavelength of light is called action spectrum. That means when we used to plot the wavelength of light being absorbed by the different pigments, then it is called absorption spectrum. And when we used to show the curve that shows the rate of photosynthesis at the different wavelength of light, listen very carefully. When we used to show the curve that shows the rate of photosynthesis, at the different wavelength of light, then that is called action spectrum. Okay, so don't get confused between the absorption spectrum and the action spectrum. Absorption spectrum simply shows the curve that the different pigments which has absorbed the different wavelengths of light. So when we used to plot the wavelength of light being absorbed by the pigments of photosynthesis, then that is called absorption spectrum. But when we used to plot the rate of photosynthesis at the different wavelength of light, then that is called action spectrum. Clear? So figure number 13.7 is the absorption spectrum or different pigments of photosynthesis, whereas figure number 13.8 shows the action spectrum. It shows the rate of photosynthesis at the different wavelength of light. Clear? I think it's clear for you the difference between absorption spectrum and the action spectrum. Now let's go through the next topic, the photosynthetic unit or the light harvesting complexes. Page number 13.11. Okay. Topic is photosynthetic unit or the light harvesting complexes. So number of pigment molecules work together to mainly release one molecule of oxygen. So we have got different types of pigments. We have got a number of pigment that is working together to carry out the process of photosynthesis, to carry out the photochemical reactions to release the molecule of oxygen. Okay, and such units are mainly called the light harvesting complexes, and such complexes are made up of three parts. One is reaction center, number two is antenna molecule, and number three is core molecules. So these three forms the light harvesting complexes. Okay, so let's discuss about each and every part. First one is reaction center. So you can see over here, it's also called the photochemical reaction center. So here you can see P700, which is mainly the absorption spectrum of the pigment of photosynthesis. That means P700 means the wavelength of light which is being absorbed by the pigment, that is chlorophyll A. Okay, so it is formed of a single molecule of chlorophyll A. So this reaction center is mainly a single molecule of chlorophyll A, and these molecules will absorb the wavelength of light, that is of either 680 nanometer or 700 nanometer. That's what it's called, 680 nm or 700 nm. Okay, so when we will discuss about the pigment of chlorophyll molecules like P700 or P680, then remember that it is absorbing the wavelength of light of 680 nanometer or 700 nanometer. So here in the figure, P700 means that the wavelength of light that is absorbed by the chlorophyll pigment is 700 nanometer. And when we will see P680, then remember that P680 means the wavelength of light that is being absorbed by the pigment chlorophyll is 680 nanometer. So we have got P700 and P680. Here we can see P700 and you can see P680. So this means the wavelength of light that is being absorbed by the chlorophyll pigment. Okay. So reaction center is mainly composed of a single molecule of chlorophyll A which will absorb the wavelength of light at 680 nanometer or 700 nanometer, and they will be represented as P680. Listen very carefully, it will be represented as P680 and P700. So P700 means it will absorb the wavelength of light of how many nanometer? 700 nanometer. P680 means it will absorb the wavelength of light of 680 nanometer. Clear? And each reaction center is mainly surrounded by about 250 to 400 light harvesting pigment molecules. Okay, so we have got around 250 to 400 pigment molecules. Oh yeah, this one. Okay, and this particular one, the pigment molecule, is divided in two categories: the antenna molecule, you can see over here, the antenna molecules, and the core molecules. Let us see what is antenna molecules means. Antenna molecules are the molecules of chlorophyll A, chlorophyll B, carotene and xanthophyll. So antenna molecules, it contains chlorophyll A, chlorophyll B, carotene and xanthophyll, and this lie outer to the core particle. You can see the core molecule here, and outer to the core molecule you can see the antenna molecules. Clear? And antenna molecules absorb light at different wavelengths but shorter than that of the core molecules. So whenever the antenna molecules will absorb the wavelength of light, it will become excited. Okay, and when become excited, when it becomes active, then the energy from these antenna molecules goes to the core molecules, and that energy and these antenna molecules will come back to their ground state. So when the antenna molecules will absorb the wavelength of light and become excited, the energy from the antenna molecules will go to the core molecule, and the antenna molecule will come back to the ground state. Now core molecules, it mainly contains chlorophyll A, chlorophyll B molecules and lies around the reaction center. So around the reaction center we will find the core molecules. Okay, and they harvest light energy directly as well as from the antenna molecule and transfer to the reaction center. That means these core molecules, they becomes active directly. They can directly absorb the wavelength of light and become excited, or it can also receive the energy from the antenna molecules. Both way, either directly or from the antenna molecules, it will transfer the energy to the reaction center, that is P700 or P680. Okay, so the light harvesting complexes. So kindly go through it. Now next one, we'll talk about the photosystems. So photosynthetic unit or the light harvesting complexes are organized into two types of photosystems, PS1 and PS2. That is photosystem 1 and the photosystem 2. Here you can see the photosystem 1 and the photosystem 2. Clear? Okay. Now PS1 or the photosystem 1 is located on both grana thylakoids and on the stroma thylakoids. So PS1 is present both on the stroma and the grana thylakoids, and it takes part in the cyclic and non-cyclic photophosphorylation. This we'll discuss in another class. Okay. So PS1 has a reaction center of a dimerochlorophyll A molecule. So you can see over here that PS1, alright, that PS1 has a reaction center of chlorophyll A molecules, and it is also called P700. So you can see in the figure that the reaction center of PS1 is called P700. Now why it is called PS00? Because it absorbs the light of wavelength how many nanometer? 700 nanometer. So PS1 has the reaction center of chlorophyll A molecule, and it is called P700. And why is it called P700? Because it will absorb the light of wavelength having 700 nanometer. Okay, and it has a proximal antennae complex of about 90 chlorophyll A and about 12 to 15 carotene molecules. So you can see the presence of chlorophyll A molecules and the carotene molecule also there, and they have got two molecules of vitamin K. Okay, and it has got cytochrome also. The light harvesting complex of photosystem 1 or the PS1 is also called LHC1. That we'll discuss in another class. Clear? Tomorrow, next class. So light harvesting complex of PS1 is also called LHC1, and it has more chlorophyll A than chlorophyll B and carotenoids. So PS1 has got more chlorophyll A compared to chlorophyll B and carotenoids. It has got the absorption or the wavelength of P700. It is also called P700 because it can absorb the light having the wavelength of 700 nanometer. Now let's talk about the operation of PS1. That we'll discuss in details in another class, but just for now briefly look over there. Then when light quanta are absorbed by the PS1 P700, energy rich electrons are emitted from the reaction center. This flows down along a chain of electron carrier to the NADP with the proton generated by splitting of water. This results in the formation of NADPS2. That means when the PS1 or when the reaction center of PS1 P700 becomes excited, then electrons from the PS1 or from the reaction center P700 will usually go down to the electron transport systems and lead to the formation of NADPS2. All this process we'll learn in details in next class. Okay. Now, hydrogen attached to the reduced NADPH is used for reduction of carbon dioxide in dark reaction. It's also called the reducing power of the cells. So the electron transport systems of PS1 and PS2 we will learn in details in the next class, but just briefly. Okay. Then we have got PS2, which is mainly located in the grana thylakoids. Clear? And PS2 is mainly composed of chlorophyll A, B and carotenoids, and chlorophyll A and chlorophyll B contents are equal in case of PS2. In PS1 chlorophyll A was more, but in PS2 the contents of chlorophyll A and B are the same. Clear? And carotenoid content is higher as compared to PS1. So PS2 consists of a photocenter. It is called light harvesting complex, which is called LHC2, and the reaction center is called P680 because it will absorb the light of wavelength having 680 nanometer. Okay, so the light harvesting complex of PS2 is called LHC2, and it's also called P680. The reaction center is called P680 because it will absorb the wavelength of light having 680 nanometer. Clear? Then oxygen evolving complex is there in case of PS2. Here also there will be flow of electrons. Clear? From PS1 PS2, which I have told that we'll learn in next class. So whenever this PS2 becomes excited, the electron will pass over cytochrome B complex, and energy passes from the protons and will lead to the formation of ATP. So here in next class we will learn about the electron transport systems from PS2 and from PS1, which will lead to the formation of ATP, which is leading to the formation of NADPS2, and will also lead to the photolysis of water or the splitting of water. So all these things we will learn in details in next class. Clear? So this much for today. See you in next class with a new topic. Thank you.